So today we're going to be focusing on oncogene addiction and oncogene targeted therapy in relation to cancer cells and the process of a cell being able to go through the cell cycle without being stopped by tumor suppressor genes because of this mutation to an oncogene. So we're going to learn about what oncogenes are today, and we're also going to talk about a few other things related to this particular topic. So our objectives for today are to be able to determine or understand what oncogenes are, talk about oncogene addiction, who discovered oncogenes and oncogene addiction. There's three mechanisms that we're going to discuss related to oncogene addiction, as well as three models of resistance to oncogene addiction or oncogene targeted therapy. And then also discuss how looking at oncogene addiction can help us find a quote unquote cure to cancer. So to start off, just as a recap, when we talk about oncogenes, it is related to cancer and cancer cells. And so what is cancer? Well, it's a genetic disease that occurs due to some type of mutation within the genes of a cell. And I say genetic because it typically not all mutations are passed on to genes, and so some people, do develop cancerous cells because of a mutation that isn't necessarily from a parent or a grandparent. But multiple or single changes can happen. It can be one single mutation that can lead to other changes to create a cancerous cell within a person's body. But typically it happens, and when that mutation occurs, the cell is able to bypass, going through apoptosis or being killed due to the mutation. The cell then grows out of control and eventually can form a mass. And so it results in multiple mutations of one's DNA. So it's a multi-stage process, carcinogenesis, and it's not just an individual effect of these activation of these oncogenes and inactivation of a tumor suppressor gene. So there's proteins that are encoded by these genes, and they have many roles and networks within the cell in which they interact. So what are oncogenes? Oncogenes are a protein that directs cell growth and differentiation within the cell. And everybody has oncogenes. And before they become an oncogene, they are what is called a proto-oncogene. And what happens is that it can occur spontaneously. It can occur by a tumor-inducing agent or some type of mutation. But the proto-oncogene is then mutated and it becomes an oncogene. And so when we look at this word and we break down this word oncology, and you look at oncogene, they have the same root word, oncos, which means in Latin mass or bulk. So we all have them, but we just don't have them in the mutated form. And the mutated forms are what cause the rise of the cancerous cells within somebody's body. So who discovered oncogene addiction in cells? Bernard Weinstein is somebody who is extremely involved in the research of cancer cells, mentioned in multiple research papers. And so he first started his discovery of oncogenes in the early 1990s. He determined this oncogene and the process of oncogene addiction to his research by studying esophageal tumors. And so in a set of esophageal tumors, his group discovered that cyclin D1 was overexpressed and diminished this RB gene expression. And these were mutually exclusive events, suggesting that there were some genetic lesions that could affect the same cell and say in the cell cycle signaling pathway. So there's multiple pathways when you talk about cancer, cancer cells, mutations, and a cell going through the process of being able to maintain being a cancer cell without it being killed. And so all these different pathways is what they were, noticing that these genetic lesions were affecting them. So his research set forth the idea of oncogene addiction and suggested that with all the variables that can affect cancer cells, that there's select genetic lesions or select genetic markers that drive a tumor cell to be able to maintain proliferation and survivability. And so this was noted and an example of this amplified cyclin D1 in esophageal cancer. And so oncogene addiction actually became a theory in 2000. So I've mentioned this oncogene addiction quite a few times already. And so oncogene addiction is where you are targeting a specific gene within a cell that's been mutated and causes a cell to become a mass because it's constantly replicating itself, going through the cell cycle. Whereas in normal cells, when you have a mutation, there are different checkpoints throughout the cell in the cell cycle where typically they can, if a mutation can't be fixed, that they can, basically the cell will go through apoptosis and be killed. But if they are able to go bypass these checkpoints and continue to replicate itself, that's when a mass is formed. And so the research done on oncogenes and the mutations that cause an oncogene to turn on indicates single dependence on a single oncogenic pathway or protein sustained for proliferation and survival. Some example of some oncogenes, these are just particular genes, RAS, MIC, and then this EGFR, which is a growth factor receptor. So when you have oncogene, when it's turned on, so the cell eventually just continues to duplicate over and over again. I mentioned apoptosis when the oncogene is off. So if a cell is mutated and the cell is not able to fix it, it would go through apoptosis. You have tumors that can form and go through dormancy and kind of hide from going through apoptosis. You have some regression that can occur. You have tumors that can be created. And so when we look at oncogenes in general, basically it's just an on and off switch determined by a mutation to the proto-oncogene. So a specific example of oncogene addiction is this chronic myelogenous leukemia, which is referred to as CML. And it's an example of this particular phenomenon. What happens is that there's a particular type of cancer that's driven by this BCR-ABL mutant oncogene. And what that is there's a piece of the BCR, which is from chromosome 22, and a piece of ABL, which is from chromosome 9. And it's just an amino acid combination. And so these genes will translocate, or basically it'll be excised from one chromosome to the other and then swapped. And so this is an oncogene that can be created whether it was in vivo or in vitro, and it led to a clinical response of this kinase inhibitor, imatinib, which targets this particular oncogene. And so this led to this reactivation of this kinase activity, which then would inhibit cellular cancer cell growth. So there's 3 models that have been proposed to explain the mechanisms of oncogene addiction beyond looking at the cellular level. So this is more of looking at oncogene addiction at a molecular level. And so there are different things to take into consideration as far as cancer-specific properties of a cell. And these 3 models are genetic streamlining, oncogenic shock, and synthetic lethality. So we're going to talk a little bit about each of these. I'm not going to delve too much into them, but I do have a little figure that can go along with each of them to kind of help understand them. So these were initially speculative. All were experimentally validated and actually can explain different but complementary facets of oncogene addiction. Not much is included on this actual research, like I mentioned in this presentation, because I want to focus more on oncogene addiction and oncogene targeted therapy, but I figured it was worth mentioning just because it helps look at this oncogene addiction at more of a molecular level as well. So genetic streamlining comes from the idea that cancer cells can undergo constant genetic drift due to selective pressures and trying to just be able to survive. And there's a lot of things that have been discussed as far as what a cancer cell has to be able to do in order to survive, not being attacked by your immune system, being able to make it from one part of your body to the next. And so genetic streamlining helps explain why cancer cells are able to survive. So cells that are cancer cells, and they're going through this genetic drift or changing their genes, being able to adapt because of selective pressures within an organism. What they do is they then will decide to lose any functions that aren't essential for the cell to be viable, to be able to adapt other ways. So this process may produce an opposite outcome, initially non-adaptive mutation that can coexist as a passenger alteration along with a driver mutation. So multiple mutations that are occurring in the genome until a new selective force, which could be a drug exposure, could cause it to mutate or change again. And the potential of this is to increase biological fitness for those cells, and also to foster emergence of resistance to clones. So being able to adapt when a new drug is exposed to those cells is what this particular model discussed. Oncogenic shock is something that's brought up in multiple research papers, and essentially it's just an imbalance of the output of the pro-apoptotic and pro-survival signaling from an oncogenic kinase following kinase inhibition by drug exposure. So most dominant oncogenes are able to sustain at the same time both pro-survival and pro-apoptotic signals. Normally, pro-survival will dominate over those over pro-apoptotic. And so here's just a diagram that is discussing oncogenic shock. So here in diagram A, oncogenes suppress growth factor receptor, that GFR, dependent signaling and addiction to activating oncogenic pathway signaling. In this diagram here, you have oncogenic shock may be predominant when that growth factor receptor is slow to reinstate after oncogene inhibition is represented, in this particular case, by the turtle. So there's no pro-survival signals in this one. In this one, you have where there's a bypass predominant when the growth factor receptor responsiveness is quickly reactivated upon oncogene inhibition. And in this one, it's obviously represented because it happens a lot quicker to be able to bypass that resistance by the rabbit. And here's just another graph discussing oncogene activation and the relationship between oncogene addiction and oncogenic shock. So in normal cells, numerous pro-survival signals predominate and keep the various pro-apoptotic stimuli in check. So these green arrows are going to represent the pro-survival signals, the thin ones, and then the thin red arrows are represented by the pro-apoptotic stimuli. So when you have these balanced, then they keep each other in check and normal cell homeostasis is maintained. However, once this pro-survival signal decreases, you then have an opportunity for the cell to exceed those pro-apoptotic signals, exceeding the pro-survival signals. Synthetic lethality is the third model, and it states that two genes have a synthetic relationship, and that is where there's a loss of function in either gene and the cell is still viable. But a loss of activity in both genes obviously can be lethal for the cell. And so the discovery of this synthetic lethality is actually what led to the discovery of the interaction between the BRCA1 and BRCA2 that deals with the breast cancer, the genetic breast cancer gene. And so this diagram here is kind of representing all three of those models. So in A, this is the genetics streamlining theory. And so again, it postulates that the non-essential pathways, which are the light gray ones here in the top, are inactivated during tumor evolution so that the dominant addictive pathways in red, this one here, are not surrogated by compensatory signals. And so above, or at the point where the signals are blocked or canceled, The dominant signals, there's a collapse in cellular fitness and the cells experience cell cycle arrest or apoptosis. And that's what happens down here. This one represents oncogenic shock. And so in this one, you have oncoproteins that trigger the same time pro-survival and pro-apoptotic signals do. And so under normal conditions, the pro-survival outputs dominate over the pro-apoptotic ones. And they follow the blockade or the process of the addictive receptor. And so again, once you're able to subvert the valence within the cell, then the cell would be in favor of the death-inducing signals, which tend to last longer and eventually, again, lead to apoptotic death. Here again, this is representing the synthetic lethality, and this is where you have basically the two genes, and as long as if one is, compromised, it's still okay. But if they both are compromised, it can lead to cell death. And this was the model that led to the discovery of the breast cancer genes. So now we're going to move on to oncogene targeted therapy and discuss what that is. And so oncogene targeted therapy is a type of therapy used to eliminate cancer cells that have identified a particular mutated oncogene. So in this type of therapy, it's tailored more to the person and studying the specific type of cancer they have and locating the mutated oncogene and targeting that mutated oncogene. And so this type of therapy is helped in target treatment plans alongside with the traditional chemotherapy. So it's a type of therapy that uses multiple medications and treatments in order to try to curb or not necessarily cure the cancer, but cause it to stop, to go into remission. So chemotherapy alone not only kills the cancer cells though, but it also kills the normal cells within the body. And so that's an issue because you don't want to kill your healthy cells, but with chemotherapy, that's more of a systemic approach versus a targeted approach. So there's therapeutic approaches such as radiation, oncogene targeted therapy, and gamma knife, which is more of a local control of killing those particular cancer cells instead of all the cells. But a lot of the times they're used, both of them are used. Chemotherapy then is used as kind of like a backup, just in case what they do use as far as the therapeutic approaches, if they happen to miss something, chemotherapy is there as a backup. And so this next slide is just a table that has the targeted oncogene, so the proto-oncogene that is mutated into an oncogene, and the type of cancer that typically is related to this oncogene mutation. And so you have the Mike, which I already mentioned. I've already mentioned this BCR-ABL. I've already mentioned the RAS oncogene. Here's Mike again. HER2, which is a type of mutation to this oncogene related to the non-genetic breast cancer. And then again, here's the Mike gene as well. So this was just some information showing you that when you have this type of cancer, that this is the oncogene that has been discovered. And so they would use oncogene targeted therapy to take care of that mutated oncogene. So like every type of medical discovery, over time you might find that there might be some resistance to that type of therapy. And so there has been resistance to oncogene targeted therapy as well. So in most cases, the targeted oncogene therapy is used to treat different kinds of cancer successfully, but there obviously have been some instances when these therapeutics have given way to relapse, or the person's not in remission anymore and their cancer reappears. So these three common themes that emerge upon resistance or have emerged to oncogene targeted therapies. The first one is secondary alterations to the oncogene drug target, and we're going to talk about each of these individually. Activating mutations in oncogenic pathway components and induction of bypass pathway signaling. And so this demonstrates that most cancers, even after targeted therapy, still retain an underlying addiction to the oncogene-induced signaling pathways. And again, there's multiple pathways. And so that's one way that cancer cells are able to work around it. So for secondary alterations of the oncogene drug target, what that means is there basically is a secondary mutation to the part of the oncogene where the drug is supposed to be targeted. And that can happen over time. So when you first discover what type of cancer the person has, and then you know which type of oncogene has, proto-oncogene has been mutated into an oncogene, you know what drugs you're going to use. They might start using that type of therapy. Well, then there might be a mutation to the oncogene that they're targeting. And so an example of this would be, so the BRCA/ABL, this can be inhibited but can result in cancer cell apoptosis. But there's a large group of patients that, when it was studied, that had showed a resistance to these existing therapies. And so what they came up with was that there was a mutation to a second site of this BRCA/ABL oncogene, and the main observed mechanism of resistance to of this oncogene and its inhibitors. So the mutation to the ATP binding pocket, referred to as the gatekeeper, is what they called it. Residue, this is a type of amino acid, threonine 315. And so what happened then with this mutation is that it would prevent this imatinib, I can't pronounce that very well, and other inhibitors from binding. So it still allowed ATP hydrolysis to occur and still was able to restore this oncogene and signaling for the cancer to continue to grow. So here's just a diagram showing the second site mutation. So this was basically what's happening is that the oncogene is reinstating the function while blocking and stopping the inhibitor activity. And it can be shown in this BRCA/ABL oncogene. And so the gatekeeper, this is that mutation that I was talking about to the amino acid. The mutagens act as an inhibitor, and it resists this mechanism. So it resists the then drug that was used to block it. And so then ATP hydrolysis is able to occur again, and the cell is able to continue to grow. So activating mutations on oncogenic pathway components, this is the second mode of resistance that was discussed. And so resistance can also occur through a mutation of alternate components of oncogene-induced signaling pathways. So if there's more than one pathway, this type of model essentially is saying that a mutation of the components for another pathway can occur. So there's multiple pathways that can aid in the growth of cancer cells, which I've mentioned, for certain types, even with the targeted therapy. One pathway might be blocked by inhibitors, but that doesn't mean that it blocks all the pathways, and so the loss of function of the one pathway can amplify the use of another one. So an example of this resistance mechanism is to this other, this BRAF inhibitor. And so there's acquisition of activating mutations and other types of signaling pathways. So while you might have an inhibitor in this particular pathway, the other pathways, these other multiple pathways, can be used. And these are all multiple pathways that drive melanoma cancer. And so here is the diagram to represent oncogenic pathway mutation. And so the mutations in oncogene pathway components can reinstate pathway signaling despite continued oncogene addiction. And so you have this alternate pathway, essentially, like I've mentioned. Here is the oncogene targeted therapy, the medication that is being used to cause the cell to stop growing or signaling and go through apoptosis. But if there's a mutation to this pathway, then it comes up with another way to work around this inhibitor and continue to grow. The third model is induction of bypass pathway signaling. So cells can be considered independent of the original oncogene through bypass of the pathway. It goes around it and bypasses the upstream pathway and goes straight to the downstream. So instead of going through all the parts, it will bypass part of it and go to a different part of the pathway. And so it works around the inhibitors, enabling the cell to still grow. So an example of this, again, is in the BRAF inhibitor, and so the RAF proteins normally function as dimers and common oncogenic mutations. They allow these proteins to activate downstream signaling pathways instead of upstream and downstream. So the bypass pathway induction, here's this diagram. So here you have call it the upstream and the downstream. And so when you typically have, again, this inhibitor blocking the hydrolysis of ATP, um, you know, initially that would cause it to want to go into cell arrest or apoptosis. But when you have this mutation that occurs, they then can bypass um, the one part of the pathway and go to the next and still be able to grow. So, the activation of bypass signaling pathways can render cancer cells independent of the original oncogene, indicated by this MET activation. And so this is a resistance mechanism for this growth factor receptor. So why has resistance happened? And so as it has been discussed, a couple other times throughout this material, there is still an Achilles heel that enables these cells to be able to still grow. So with all the different genetic and epigenetic abnormalities, it's really hard to identify all the characteristics of a cell, essentially because they are constantly being able to mutate and change their molecular structure. And so with all these genetic and epigenetic abnormalities in tumors and the heterogeneity of the tumors, it's hard to determine the specific state of an oncogene or of oncogene addiction in some cancers. So there's not methods to fully assess everything that controls cell proliferation, differentiation, apoptosis in a normal cell versus a cancer cell. But there are definitely approaches that are being used to help identify this Achilles heel in specific types of cancers. And so they'd be able to use it. One specific example would be to use small interfering RNAs. And so why we would focus on RNA is because RNA is the nucleic acid that is used in transcription and translation to create a protein. And so proto-onco genes are proteins that get mutated to turn into oncogenes. So by using the RNA to identify which genes require to maintain proliferation and signal specific types of cancer cells, we then can use that to identify the types of drugs that can be designed to target the related proteins. So where do we go with oncogene addiction and determining the future of cancer with this type of research? So with the discovery of oncogene addiction by Weinstein, it's enabled this one-step remedy for some type of cancers, but not for all. And we also know that there have been, throughout this presentation, we've discussed mutations and mutations that are enabling the resistance to oncogene targeted therapy. And so for some cancers, it will work. For some cancers, there's going to be multiple steps that are going to have to be used in order to arrest cancer cells and for a person to go into remission. So what do we need to do for the future of oncogene targeted therapy and studying oncogene addiction? Well, there are multiple things that were discussed through some research papers, but one thing that I think needs to be done would be to genetically identify all types of cancers. And again, that's just a really hard task to do just because of the mutations that can occur. We can determine a type of cancer based on where the cancer cells originate. But by genetically defining all types of cancers, we can move forward into a one-step remedy, hopefully, with, not necessarily curing cancer, but being able to cause it to go into remission. One thing with cancer is every time I feel with the research that's been done is once some ground has been made, like researchers take one step forward with what they've discovered or a new therapy, and then it's like cancer takes two steps forward. So they're always a step ahead in terms of mutating. As far as the future of oncogene targeted therapy, the therapeutic resistance needs to be addressed earlier and maybe make better inhibitors based on what has been discovered so far. And so these better inhibitors can help decrease the chances of being resisted and still have the same outcome in the end as it has for some that have had that one-step remedy. So is this the missing piece to what we call the cure in cancer? And I would say it's not the missing piece, but it is part of the puzzle that's helping scientists figure out how to come up with a cure for this disease. So it has come a long way from the original scientific acknowledgement of cancer and different treatment plans, but there's still a lot to learn. But oncogene addiction and oncogenes was a huge step for that. The discovery of oncogene addiction was one that really enhanced the targeted therapy idea, making it a person-specific therapy versus a one-size-fits-all treatment. And so this is going to enhance the treatment plans in the future for people that have cancer. And so, like I mentioned, there's not a one-size-fits-all treatment, but oncogene targeted therapy resistance has shown us that. And so at the present time, the best molecular targeted agent and the appropriate combination of therapy for a specific cancer has helped with this. But with rapid development of molecular targeted agents and research enabling us to learn more about oncogene addiction and targeted therapies, I think it gives scientists a one-up on trying to find that cure that they had been looking for. And so just to recap, my slides are messing up, of the main points. Recap, so we went over what oncogenes were, oncogene addiction, who was responsible for discovering it, the mechanisms of oncogene addiction, and the models of resistance of oncogene addiction, and how studying oncogene addiction will help find a cure to cancer.